So next, we have the Dr. Zhang as our third speaker. So Dr. Zhang, are you here?
Yes, yes.
I'm not hearing you. Okay, okay.
Can you hear me?
Okay. So my player, yes, I can. So my great pleasure to meet you, Dr. Zhang. Yeah, nice to meet you. Okay, let me introduce Dr. Zhang. He is a senior director of the Economics APAC. Dr. Zhang has his PhD degree from the Uterine University and his postdoctor at the University of the Maryland, Memorial in the microbiology. Dr. Zhang leads the Igenomix APAC team to introduce ERA to Asian in 2017, and his by far has accumulated over 20,000 ERA cases from the 313 IVF clinics across the APAC. Today, Dr. Zhang will share the APAC insight of the endometrial receptive with the EndomeTRIO data. Now, Dr. Zhang, it's your return. Welcome.
Okay, thank you very much for the introduction. My name is Andy Chang. Now I'm based in Tokyo. I'm talking from Tokyo, and thank you very much. Let me share my screen. Okay. You know, I got a short notice that I have to talk this morning, so please forgive me if my preparation is not very complete. Today I would like to talk about this, the application of the endometrial in the Asia Pacific region. We talked about this, the solving the puzzle of endometrial receptivity when we try to introduce the endometrial. So again, my name is Andy Chang. I'm the Regional Senior Director of Igenomix in the Asia Pacific region. Thank you very much for joining this session. So globally, the live birth rates in ART ranges from 25% to 30% in every initiative cycle. And the most fundamental reason is that the implantation is a big issue, right? So as you can see that this is adult data from the Netherlands in about 10 years ago, and 74% of the failed IVF cycles equal to be the failed implantation. So implantation is a big issue when we perform the ART. So the contribution of embryonic and endometrial factors, you can see that in the Drosophila or mouse, there's almost no aneuploidies. And in human, according to the age, there could be from 20% to 100% aneuploidies happening. And implantation rate, you can see that this is for human. That for natural cycle, usually the implantation rate is 35%. And if you do PGTA with euploid embryos, it could increase it could be up to 60%. But for rodents and rabbits, the implantation rate is always very, very high, like 95, 96%. Why? Because they have a different strategy. That rodents are using a strategy called embryonic diapause, but human needs goes with the decytosization. So when the embryo comes into the uterus, the first thing it needs to encounter is the microbiota in the environment. And so these are the studies that we have done, including the basic research throughout the implantation process. And you can see that when a embryo enters into the uterus, the first thing it encounters is the microbiota, and then the embryo is going to have a crosstalk with the mother, maternal embryonic crosstalk using some microRNA messengers, which is also by our study, we found that microRNA are coming back and forth the embryo and the maternal body to convey the messages. And then it comes to the receptivity part, that whether the endometrium has the receptivity or not, and then goes through the decytosization and start to develop the placenta and then the fetus, right? So this is the whole process, and you can see that very important things here are the microbiota and also the window of implantation, or the endometrial receptivity right here. I would like to cover I would like to cover these two parts, which is the microbiota and the window implantation today. So as you can see that there are factors to be considered, but however, the traditional method are only considered as histology. These are more visual methods that we have been using, hysteroscopy or ultrasound. These are our visual methods. And we, Igenomix, develop different methods, including the omics using the transcriptomics, which is ERA, and also we developed the sequentomics, which is non-invasive ERA that we try to extract the endometrial fluid to make it more non-invasive, right? And also we developed a treatment using stem cell to treat the Asherman syndrome. And also the last part is another topic, which is microbiota, that we developed two tests called Emma and Alice that covers this part. So combining the then technology and the now technology, we might have a more complete or thorough view of the endometrium. So the first thing is the endometrial receptivity analysis. I think Dr. Kasala and Dr. Lee have introduced quite a lot about this test, so I will skip some basic part. But I would like to give you some refreshment of some basis of this or some theory of the ERA test that while we developed. I think Dr. Lee also showed this, so I would not spend too much time on this. But important thing is that we in the beginning of the cycle, if it's a natural cycle, estrogen will start, concentration will start to raise, and then ovulation, and then progesterone concentration starts to raise again, and then prepare for the implantation. And you can see the thickness of the endometrium also changes accordingly. And for HRT cycle, as you can see that it's administered by artificially, right? So it's the patients are given the estrogen and also progesterone, and you can see that after almost 2 weeks of estrogen treatment, progesterone was administered, and the first shot of progesterone is called P+0, and after about 5 days, it's basically P+120 hours. It is considered to be the standard timing of the window of implantation, right? So we consider this is a more common practice for most of the clinics. And if you take the biopsy here, the ERA will tell you that, okay, this is a receptive result. If the patient has a standard window of implantation, yes, it is like that. However, some patients have a shifted or displaced window of implantation. For example, if the window of implantation is displaced to a later time or delayed, we call it a delayed window of implantation. If you take the biopsy still on day 5, you will find it the result to be pre-receptive because the window is not open yet. So that's before the window of implantation. And some patients might have a window advanced to the earlier time. And if you take the biopsy on P+5, it will show a result called post-receptive because the window is closed. So according to the result, we are able to predict the window of implantation of each patient. So for example, if you take the P+5 biopsy and found the result called pre-receptive 2 days, that means the patient needs 2 extra days of the progesterone treatment. So we suggest that P+7 is supposed to be the window of this patient. And if that's P+5, if the result shows pre-receptive 1 day, then you can have an extra day for progesterone to make it P+6. And on the other side of it, if you have a post-receptive, and then we will suggest a 1 day earlier biopsy again, because post-receptive is less predictable. So the theory in back of this is that the endometrial, the progesterone concentration will trigger the expression of 236 genes, which express differentially throughout the process, the window of implantation process. Okay, the period too. So we can see that in the proliferative stage and also pre-receptive, receptive, and post-receptive stages, different genes are expressed differentially. So according to these profiles, we are able to define that the patient's sample or the biopsy is in which stage. Okay, so this is the theory back of this. And we suggest that recurrent implantation failure patients, there's a big portion of them can be that ERA can help them, right? And also, or patient at advanced ages, they don't have a lot of embryos to transfer, they don't have a lot of chance, and we suggest them to use this ERA. And also the test cycle needs to be a mock cycle. That means that you're not able to do any embryo transfer. So they need to be done in two separate cycles. So that's some kind of disadvantages that might be considered. Okay, so these are the papers that we have published during the development of the study. And we are using the next generation sequencer, sequencing 236 genes. In the beginning of the study, we started a whole genome or all the genes. We actually screened all the genes and found that only 238 genes are involved in this process, and we further decreased 2 genes that are not very relevant. So now we're using 236 genes for the profiling of the window of implantation. And now there are more than 500 clinics in the Asia Pacific regions using this one, and more than 300 clinics in Japan are using this test. And as I just explained, that in each stage of the implantation of the window, you can see that the different gene groups are expressing. Okay, so according to the expression profile, we are able to predict that the patient sample is in which stage. For example, pre-receptive or receptive or post-receptive, and we are even able to tell the doctor that the sample or the biopsy is 1 day earlier than the window, 2 days earlier in window, or sometimes it's 12 hours earlier than the window. It could become very precise because we accumulated a lot of samples and trained the algorithm to predict this test. So this is the idea or image that you can think that, you know, we use the statistics or informatics method to predict that each sample, and we can see that each the sample group, they fall into clusters, and you can see that if the sample falls into the proliferative, that means the sample is really too early, and then probably not really exposed to progesterone, and then we can try to dig out the problems. And also you see that different clusters, and when the samples falls into different clusters, we use the clusters to classify whether the sample is receptive or non-receptive, or early receptive or pre-receptive. Okay, and the timing for the endometrial biopsy, sorry about the typo here, that we were able to do natural cycle and HRD cycle, but we do suggest HRD cycle. As I just said, that progesterone induces the expression of these 236 genes. So if the progesterone concentration is not stable, or I should say not reproducible, then the result will not be very reproducible. So that is why we suggest the HRD cycle for ERA. And natural cycle, you can do that as well. If you want to do natural cycle, basically the timing of the biopsy is LH surge plus 7 days. Okay? But as I just said, that, you know, the internal secretion of the progesterone is not stable each time. So we would suggest a injection of HCG, and starting from the SCG 7 days after that will be the biopsy time. And for HRT cycle, we will suggest the estrogen in the beginning, and followed by the progesterone treatment. And the first shot of the progesterone is P+0 after 120 hours is our biopsy timing, right? And before the progesterone treatment, we suggest you to test the thickness of the the endometrium, we usually would suggest some lining of 7 millimeter, and also we suggest you to measure the endogenous P4 to be less than 1 nanogram, because if we are doing the HRT cycle, it needs to be very much controlled so that we can have the very good reproducibility. This is the concept of window of implantation. That we actually found that the window of implantation, most of the patients have about 24 to 36 hours, and the average is about 30 hours of the implantation. And in some very specific cases that we saw, patients might have some window as short as 12 hours or so. And some patients might have a long window, like 48 hours or so, but the average is about 30. And you can see that if the biopsy shows receptive, that means it falls into the middle of the window. And actually, you can see right here that early receptive and also late receptive, they are also in the window. However, early receptive is in the very beginning stage of the window, and late receptive is in the late stage of the window. That means they could, if you transfer at this timing, the embryo might implant. However, it's not going to be very stable, and the patient might suffer from the biochemical pregnancy. So that is why we try to bring the transfer timing to the very middle of the the window. Okay, so that is the concept of this test. And for the clinical data that we have done so far, we found that about 30% of the patients we have tested are non-receptive, and among them, most of them are pre-receptive. So this is quite interesting data that for patients that have a displaced window, most of them are delayed cycle, delayed windows. So they are the result shows pre-receptive, and very few of them shows post-receptive. That means 9 to 1. Only 10% of the patients that are non-receptive tend to have a advanced window of implantation, and very few of them shows a very early stage called proliferative. So this is probably something wrong with the progesterone treatment, but that could happen too. Okay, so and among the receptive, actually, as I just showed, that even they are in the window, the biopsy shows that the sample is in the window, the patient's P+5 is in the window. They might not be in the middle of the window. Some of them are early or late receptive. They are on the edge of the window, so they need to have a small or fine tuning of the transfer timing. And we suggest a 12 hours change of the timing. All right. So comparing the Asia Pacific data with the world data, that in Asia Pacific region we have done more than 20,000 cases, and you can see that the data are very similar. So so regardless of the races or the countries, the data are very similar, and you can see the ratio of the so-called receptive is about 42 or 43, 44%. That means there's no fine tuning, not even fine tuning needs to be done. And for those that are early receptive are 11%, or late receptive 16%, very similar, very similar. And for pre-receptive 1 day, 24% and 27%, very similar. So we do not see we do not see the difference between the world data and our Asia Pacific data. And the patient age distribution. This is very interesting data that we have actually found that for those 20,000 samples that we have tested, you can see that most of the patients fall into 35 to 44 years old, and many of these patients are Japanese patients. So because the patient's age in Japan is higher than other countries, so maybe this is something that might be different if we see the world data. And something interesting to see here is that for higher ages, for patients with higher ages, they tend to have a delayed window of implantation. So you can see that we for patients that have much higher ages, they have more pre-receptive results. So they have a delayed window of implantation. So we found that observation. However, we haven't got the significant data analysis yet. And I think this, I think Dr. Casala and Dr. Lee also introduced about this trial, so I would skip this one. And you can see that for pregnancy rate and live birth rate, or cumulative pregnancy rate and cumulative live birth rate, our personalized embryo transfer does contribute to the higher clinical outcome, a better clinical outcome. So you can see that we have 6 patterns of the results that you can see. As I just said, that receptive means that the biopsy timing is exactly right for this patient, and early receptive, you need to have a 12 hours of more progesterone treatment, and for late receptive, you need to reduce 12 hours of the progesterone regimen. Progesterone treatment, and for pre-receptive 1 day, you need to add another day of the progesterone treatment. And also for this one, for pattern 5 and 6, because pattern 5 means that the patient's biopsy is way earlier than the window, so we would suggest a second biopsy to do the confirmation to make it very precise. And post-receptive, because as you might have seen, that the gene groups that are expressing after the window are basically preparing, like apoptosis or inflammation groups. They're preparing for the period. They're preparing for the next period. So even post-receptive 1 day or 2 days, the profile looks very similar. So it's very hard for us to predict whether this is post-receptive 1 day or post-receptive 2 days. So that is why we suggest a 1 day earlier second biopsy to confirm whether the biopsy can pass into the window or not. All right. So at last, I would like to raise a few questions for discussion that we also received these questions from different doctors. So is the window of implantation changeable? In what conditions might the window of implantation change? That the BMI, we found that a BMI change, especially for those patients that have BMI more than 30, that has more non-receptive result. And also endometrial curettage and or giving the birth. Some doctors told me that the endometrial curettage or giving the birth might change the window of implantation, and some says not. So there's a paper that was published by Carlos Simon. It's a collaborative research with Harvard University. Sorry, it's a Stanford University, showing that each the research was on the single cell expression analysis, and it shows that when we are taking a biopsy, we're taking all different cells, including epidermal cells, cells or soma cells, and all the different layers of the cells are taken as a biopsy. And so if the composition of these cells change, that could also cause some change of the window of implantation, right? And how about chronic endometritis? Does it change the? Because Dr. Kuroda in Japan also reported a case that very heavy endometritis actually affected the change of the window. So that could also be something that we can consider. And also drugs with functions related to uterus or operation in the uterus, yes, that could change the composition of the cells. That could also change the window of implantation. So without these, our report shows that our study shows that the window of implantation, at least for 3 or 4 years, it should not change. Okay, and the second question is that does ERA work for day 3 embryos too? Theoretically, yes, and we have doctors that are using a result for day 3 embryo transfer. However, this test was developed for Brussels cyst, and for day 3, if the embryo developed in a very stable way or speed, then theoretically the result can be used. If you subtract the result by 2 days, it's supposed to be useful for the day 3 as well. And is your ERA result compatible between the natural cycle and HRT cycle? As we just said, that HRT plus 5 is the timing of our so-called standard window of implantation. But for natural cycle, LH plus 7 or HCG plus 7 is we so-called the standard window of implantation. And do you think, if you have the result called the HRT plus 5, which is receptive for one patient, and can we also transfer the embryo LH plus 7? It's a totally different cycle. Okay, and our study showed that most of them are translatable, or they are compatible. However, it's not absolute. We saw some exceptions as well. So this is something that we need to consider. It's not always compatible between these 2 cycles. Okay, and because of the time, I need to move on to the ERA and Alice. Okay, and we have so we consider that the bacteria in the endometrium and the best status of the endometrium is that the optimal is there's a lot of Lactobacillus, and then the dysbiotic bacteria is that there's not enough Lactobacillus, and the worst case is that pathogenic bacteria, there's not enough Lactobacillus, but there's too much. There's some pathogenic pathogens in the endometrium. So we did some pilot study and found that the uterine cavity is not sterile, and also endometrial microbiota is not a carryover from the vagina. They have different profile of the bacteria. And also the percentage of Lactobacillus together with the specific pathogens are significant variables to predict reproductive success. And Lactobacillus prevents the vagina from inflammation because the Lactobacillus generates the lactic acid and make the pH to be lower than 14.5. So this is the mechanism in the vagina. However, in the endometrium, it could be different. That we found that because of the pH is not as low as vagina, it's 4.5, and there's some possible function of the Lactobacillus in the endometrium can be considered. For example, generating lactic acid is one of them, and also it could also activate Th17 cells, also promote some functional differentiation of the dendritic cells, and also generate some H2O2 bacteria, as well as some other function that we haven't found. So molecular assessment of the endometrial microbiota by 16S rRNA sequencing is that the test that we developed. We extracted the DNA and then sequence the 16S rRNA gene. And these are the 2 papers that back up that are the basis of our Emma and Alex test. And the paper on the left, you can see that for the group of live births, there's a lot of Lactobacillus. And then the group of the miscarriages, there's a lot of Gardnerella. And then the group of the implantation failure, there's a lot of Streptococcus, which is the pathogen that causes the endometritis. And on the right side is we try to compare the traditional ways of the CE diagnosis and with the 16S rRNA sequencing, and found that none of them is perfect. But our Alice could be an option for diagnosing the chronic endometritis. So Emma, as result, you can see that Emma tells you that the percentage of the Lactobacillus, 90% is our standard, right? So if the Lactobacillus is not more than 92%, more than 90%, it is normal and this is a very good optimal result. And if it is not, but there's no pathogen, so pathogen is negative, there's no treatment. Okay, so if it is more than 90%, it's Lactobacillus dominant and it's very good, you can transfer. If it is not, then you will need to have a probiotics treatment, right? But if the pathogen is positive, if the ALICE is positive, you need to treat the pathogen with the antibiotics that we suggested in the report, and plus followed by the probiotics treatment, which is vaginal probiotics. Right? And also there are some other results called the mild or ultra low, because in Asia there's a lot of antibiotics treatment and they use the wide spectrum antibiotics. So you see a lot of endometrium with a low, very low biomass. Right? So the result of pattern 4 or 5 doesn't mean that our test was a failure. We do have a control of the DNA, because when we take the biopsy, we have the DNA from the endometrium as well, which is the human DNA, as well as the bacteria DNA. So we do quantitate both of human DNA and our bacteria DNA. But in some cases that we could see that the control DNA, which is the human DNA, but we are not able to see any bacterial DNA. In that case, the test itself is valid. However, we do believe that the biomass of the bacteria is very low and is almost no bacteria in the endometrium. Right, so these are the available vaginal probiotics in Asia Pacific. In Japan, they use the Invac from Poland, and this is called Lactoflora from Belgium, and also in Taiwan. In China, they have different bacteria probiotics as well. And for thousands of the samples, we've seen that in addition to Lactobacillus, Gardnerella is very common in the endometrium, which is not good, which is not a good bacteria. And also, among those pathogens, Streptococcus is most common pathogen that we detect in the endometrium. In that case, ALICE is positive. Right? So this is also the clinical data of MI-ALICE. That normal 25%. This is the average data, right? And abnormal 24%, ultra low 30%, mild but dysbiotic is 19.5%. You see that these two shows that the bacteria in the endometrium is very low, the biomass is very low, and you see that actually in Asia there's a lot of use of antibiotics, probably not only in Asia, but these wide spectrum antibiotics will kill a lot of good bacteria as well. All right, so in Japan, from 18 to 21, we started a research with a group of ART clinics called the GSART, and we use the MI analysis, try to see the efficacy of that, and also we want to see that after the treatment, can the Lactobacillus level increase in the endometrium or not, and also according to this treatment, can we improve the pregnancy result or not? And we have done more than 1,000 samples, and now we're waiting for the result to be published. All right, so some questions to be asked. Is the species of Lactobacillus important for the endometrium? So far, we are not sure. There's some paper talking about the Lactobacillus in the vagina. There are different species, like for example, Lactobacillus gasseri, Lactobacillus iners. These are the species. But however, although we only sequence the bacteria to the genus level, and also we are supplying the probiotics to the genus level, it looks like they are working quite effectively, and then also the clinical outcome of these Lactobacillus are quite good. So we are still trying to see or do some more research on this aspect. And is oral treatment effective or increasing the Lactobacillus in the endometrium, which we don't know. We found that because the oral treatment is not directly going to the endometrium, it takes more time. And if you are the oral treatment is not probiotics, it's prebiotics, it takes even more time. So that is something that we don't know. But for those vaginal probiotics, we do see the effectiveness and efficacy, which the doctors have been have proved, right? And so we are we also ask many questions like this. Called, do you think CD138 is also important, or only ELIS is fine? The nature of the ELIS test is that it's testing the pathogens, right, the bacteria. But CD138 is testing and diagnosing the status of the inflammation. So basically, these are different tests. But if you know, even if you have a positive result of CD138, you are giving the patient antibiotics, which is wide spectrum. You don't even know which pathogen is the cause of that. But ELIS gives you the right, very specifically what pathogen is inside the endometrium. So that could give you a very direct result of that without killing other good bacteria. Okay, so also the C affects the window of implantation. In very, very serious cases, yes, but those cases could be seen by hysteroscopy as well. So most of doctors, they use a hysteroscopy in the very, very first visit. So we believe that, yes, it could be possible, but it's not, but it should be limited in the very serious cases. So this is the concept of the endometrial. We have a ERA that tells you the timing of the window of implantation, and EMA tells you the Lactobacillus level, and ELIS tells you the pathogen. And combining all these, you can use one sample and then generate all the results. Okay, so this is the microbiome team and ERA development team. And okay, thank you very much for your time. Thank you.
Thank you, Andy, for sharing the comprehensive data and all insights. I think with all the academic exchange and the collaboration, we can work together to maximize the chance of the pregnancy by EndomeTRIO guided PET for IVF patients. For now, Andy, we have received some questions. So the first question is, for ERA, do you think using aspirin will impact ERA or ELIS or EMA's results?
Did you say aspirin?
Yeah, aspirin.
Well, we receive a lot of questions about like this, right? But the aspirin is not really functioning on the endometrium or the uterus. So basically, like people ask questions like cold medicines or other things. So basically, we have a list of the drugs that might or might not affect the result of a window of implantation. But basically, we think that aspirin is not some drug that might affect.
Okay, thank you. So another question is then for the probiotics, what do you think be more effective, very vaginal or oral approach is more effective?
For the treatment of for the microbiota in the endometrium?
Yeah.
Yes, as I just mentioned, that oral is not effective because the most direct and straightforward way is using the vaginal probiotics, and we actually proved that in the GSAR study we also have seen that the increase. We did a second test to actually verify whether the lactobacillus level is has increased or not, and we did see that at least 70% to 80% of the patients have an increased level of lactobacillus in the endometrium. However, not everyone. So we're still thinking that whether these patients that, with the first or second treatment of the probiotics, even with the treatment of probiotics, the level is not increased. That might be because I think the reason is probably because of the species. That could be some kind of the reason. So some doctors, they try to use a different probiotics, and then they found the increase of the lactobacillus. So there's a lot of things that we still have to research.
Okay. Thank you again for all the answers and the wonderful presentation.